Remember, please get your parents' consent before attempting any activity.
Hey y'all, this is Miss Rouse, the Activity Boss. I'm here with Bgcsa Live, your virtual Boys and Girls Club, and today we're going to do a really cool science activity where I'm going to teach you how to extract the DNA out of a strawberry using simple household materials. And you're also going to hear from a special guest, a professor of genetics and biology at the University of South Alabama. Her name is Dr. Mary Kretz, and she's going to explain a little more about DNA, what it does for the strawberry, and why it's so important. So today's episode is all about DNA. And DNA is found inside the cells of every living thing, and it's very important for us and every living thing on this earth. So DNA is found inside cells, and the cell is the smallest unit with the basic properties of life. So if you took this strawberry and you divided it into two, it'd be smaller. And then if you kept dividing it and dividing it and dividing it till it was so small that you could only see with a microscope, you would be able to see cells. So cells make up everything. And DNA is found inside of the cell. So cells are inside strawberries, they're inside you, they're inside a cute puppy, everything that is living. So humans are made up of tons of cells, about 75 trillion. And there are other types of organisms like bacteria and yeast that are actually only made up of one cell. DNA is the material that carries all the information about how a living thing will look and function. DNA inside of humans determines things like what your hair color will be, what your eye color will be, how tall you will be. DNA is kind of like an instruction manual. It tells our cells to make different kinds of proteins. And it's also kind of like a secret code. So the sequence of the different parts of our DNA determines what we will look like and how we will function. So the sequence of DNA is what makes different living things unique. It's what makes this strawberry bright red and kind of squishy, and it's what makes carrots hard and orange. So DNA is really important and plays a very important role in living organisms. So you're going to hear from Dr. Kretz, who's going to explain a little bit more about that and why scientists are so interested in DNA. But first, let's get started doing our simple at-home DNA extraction. To do this activity, you need 2 teaspoons of dish soap. You need a jar or a glass, and then you need either paper towels or a coffee filter. It actually works best with a coffee filter, but I just had paper towels, which works as well. A jar or glass or cup to put liquid inside and stir. A plastic baggie. Cold rubbing alcohol. This is really important and needs to be cold. It helps with the reaction. So you can put it in the fridge or in the freezer before you do this. A teaspoon of salt, something to stir with, half a cup of warm water, 1 strawberry. I cut mine up a little bit, which helps, but you don't have to do that. And a wood skewer or a toothpick or a fork, something where you can put it inside of the jar and scoop something out. So we're gonna start by making our solution. So you're gonna take the half cup of warm water and pour it inside jar or cup, half a teaspoon of salt, and 2 teaspoons of dish soap. You're going to want to mix it up well to get the salt to dissolve. You can put that aside for a little bit. Take the strawberry, put it inside a plastic baggie, and then mush it up. And you want to mush it pretty well. You don't really want there to be chunks. going to take 3 teaspoons of your solution, pour it inside your plastic baggie. You want to mix that up. You don't want it to get super foamy, but give it a good mix. And what the dish soap does is it helps to break down the membrane that's around the strawberry cell. And so the DNA is found inside the nucleus, inside the cell. And so we're trying to get the DNA to come out of the nucleus. And pour it on top of the jar. So either into your coffee filter or into your paper towels. And then you're gonna let that sit for about 5 minutes. And you can see that it's starting to just drip through. So you're gonna let that sit for 5 minutes, and the liquid is going to seep through the paper towel or coffee filter. It's gonna leave the chunks out. So some of our solution in strawberry has dripped through. So you don't need too much of the liquid really to get some DNA out of it. But what you can do is very carefully take your coffee filter or paper towels and then just squeeze so you can get more out. But you have to be careful not to break the paper towel bag or the coffee filter bag, because you'll get other stuff inside of this liquid that you don't want in there. So we're trying to isolate different parts of the strawberry. All right, so I have some liquid, not too much. So if I had let it drip even longer, I would have had more. So if you want to collect more DNA, you can be more patient. And I also used a thick layer of paper towel. I would actually use a bit of a thinner layer so it drips through better, or a coffee filter. And then you're gonna take your cold rubbing alcohol, and then you're gonna get down so you can really see the level of liquid that you have. And what you wanna do is pour it down the side, the rubbing alcohol, and I'm gonna pour in an amount that's about equal to the amount of the strawberry liquid. So I'm just gonna pour in a teeny little bit. It's not an exact amount that you need. A little bit more. So everything that we used played a different role. So the soap helped to break down the cell membrane. So there's a membrane around the cell that helps to protect the cell, and the DNA is inside of the nucleus, which also has a membrane around it. So the soap helped to dissolve those membranes and get the DNA to come out of the nucleus and then out of the cell. The salt helped to release the DNA strands.
Use a toothpick or skewer to scoop into the jar. The snot-like pink strand is actually clumps of strawberry DNA.
So hi, my name is Mary Kretz. I'm a scientist. I'm both a geneticist and a developmental biologist, and I work at the University of South Alabama. So to get this job, actually what I did is I did a lot of time training to do different research. And so I did a PhD at Yale University, and then I went to the University of Minnesota to continue some work in the research. And then from there, I now am at the University of South Alabama.
So we're working on extracting the DNA from a strawberry. And could you explain to us, from a scientist's perspective, what is DNA and what has DNA?
Sure. So everything alive has DNA. If that's how it can live. And so what DNA kind of is it's everything's instruction manual. And so, for example, like if you went to the store to get a bunk bed, then you're going to get an instruction manual to put that bunk bed together. And so its legs go here, ladder goes here, beds go here. And so that's what the DNA does too. It's in all of our cells, in anything that is alive in their cells, telling it what to do.
So what does it do for the strawberry? Why is it so important?
So it gives different, so each of the cells have the DNA in them. And so what's happening is different things are being made from that DNA. It's telling the strawberry to do things. So it's telling it to taste the way it does, because what happens is it ripens in a certain time. It expresses different things that makes it taste sweet. So the reason why we like strawberries is because they're sweet and they're expressing certain things that are sweet. And it responds to the environment to ripen at certain times, etc. And so the DNA is kind of controlling it to be red, to be sweet, all the things that we like about a strawberry.
So what can scientists do with DNA?
So for example, with strawberries, actually, there's also wild strawberries, and they're a lot tinier, and so they taste a little sweeter. And so scientists were trying to understand, why does that strawberry in the wild taste sweeter than the ones that we have? And so they can look to see what's different in their DNA and what different things are expressed in the cells. And so they can compare those. And so that's some of the things they do. But then also in us, because everything alive has DNA, what they can do is they can say, what's in our DNA that is expressed, and if something is wrong about or different in the DNA, then that's how we often get diseases.
So what would happen if I ate the strawberry DNA? Do you think I'd become like a weird strawberry mutant, or what would happen?
That's a great question. So actually, unfortunately, we don't become strawberry mutants, or maybe fortunately, we stay as ourselves. But what happens is because we're eating all kinds of things that have DNA in our daily lives. So if you eat strawberries, they have DNA, and we just consume the DNA in the strawberry. Or if you had a hamburger, that beef is going to have DNA in it too.
If you planted it, like the DNA we extract and put it in the ground, do you think it would make a strawberry?
No, unfortunately it won't, because what the DNA needs to be surrounded by a cell. And so if it doesn't, and it needs to have the different components that a cell has. So if it's just alone as DNA, it's not going to do anything. But if you have that seed and you plant that seed, because that seed has the components it needs, then you could get a strawberry if the conditions are right.
Check out our other virtual club videos on Facebook and YouTube by going to @Bgcsa. Also, make sure to subscribe to our channel and like this video. You can also share it and let us know what you think about it. Also, make sure to check out our summer camp, Camp Chandler Tonsmeyer, for some fun social distancing. You can sign up by going to bgcsaal.org/camp. See you next time.